Hello, my name is Zeira and I'm a postdoc with Professor Ben Raphael at Princeton University. I would like to thank the organizers for the opportunity to present our work on copy number evolution with weighted aberrations in cancer. Cancer is an evolutionary process where somatic mutation accumulates in a population of tumor cells. The clonal composition of a tumor may change over time or in response to therapy, and thus accurate quantification of tumor heterogeneity is important for understanding the progression and the effective treatment of cancer. Measuring the mutations in extant clones aids in reconstruction of tumor history and the identification of mutations that drive cancer proliferation, therefore providing insights into the mechanisms of cancer progression and metastasis. Mutations in cancer range in scale from small, single nucleotide variations in intels to copy number aberrations, or CNAs, that delete or amplify large genomic regions. CNAs can scale from a few kilobases to whole chromosome or chromosome arms, and even the entire genome. CNAs are a common type of somatic mutation in many cancer types and are the most common mutation in some of the cancers. CNAs play an important role in the driving cancer development, and thus characterization of these events is essential for the disease diagnosis, prognosis, and treatment. CNAs provide important information for reconstructing tumor evolution. Recently, single-cell sequencing has been emerging as a promising approach for assessing tumor heterogeneity and evolution. Therefore, there has been a lot of interest in modeling copy number aberration evolution lately. Modeling this evolution requires us to define how one genome evolves into the other due to CNAs, or how two genomes are similar. However, there are a few major challenges in modeling copy number evolution. First, genomes containing multiple duplicated regions are difficult to correctly reconstruct from current short-end DNA sequencing technologies. Second, even with a reconstructed sequence, most genome evolution models that allow multiple genomic copies are computationally hard to solve. To address these difficulties, recent research is focused on modeling the evolution of copy number profiles, a simplified representation of a genome. A copy number profile, or CNP, is a sequence of integers that indicates the number of copies of each region from a reference genome that are present in the genome analyzed. Thus, copy number profiles only model the number of copies of each segment of the reference genome and not the sequence of the arranged segment. However, they are a useful representation because they can be readily derived from multiple technologies. Copy number profiles can be derived from DNA sequencing data of bulk tumor samples using specialized algorithms that infer the profiles from the mixture of normal and cancerous cells in this data. While earlier technologies calculated the total copy number of the two alleles, recent methods are able to derive haplotype-specific profiles. Single-cell sequencing precludes the need for deconvolution of bulk samples into integer copy number profiles, and recent methods are able to derive profiles for each sequence cell separately. Modeling copy number evolution using copy number profiles is still challenging. Some of the methods that have been introduced to describe the evolution of copy number profiles treat each segment independently, for example using Euclidean distance. However, unlike single nucleotide mutations, copy numbers of consecutive segments are not independent. Other methods try to model copy number variation events. The fish tree model, proposed for analyzing copy number profiles derived from fish experiments, includes single position changes as well as whole chromosome and whole gene duplication events. However, this model lacks efficient algorithm to compute the distance between profiles, and thus applications of this model have been limited to very short profiles. An alternative model of copy number evolution is the copy number transformation model. We start by presenting the copy number transformation model and its limitations. In the copy number transformation model, a profile S evolves into another profile T by a series of copy number events called a transformation. Each event can either increase or decrease the copy numbers of a contiguous interval in the profile by 1. We refer to these events as an amplification and deletion. However, zero entries are not affected by any events, meaning that a lost segment can never be regained. For example, copy number profile S can be transformed into T using 1 deletion and 2 amplifications. The copy number distance, or CND, between two profiles is the length of the shortest transformation from one profile into the other, in this example, 3. Notice that the distance is not symmetric, as there is no transformation from T back to S. MEDIC was the first algorithm to compute the copy number distance. It uses a heuristic to reconstruct a phylogenetic tree that minimizes the total copy number distance from extant cloned copy number profiles of a tumor, and it has been successfully used in several cancer studies. We showed that a copy number distance between a pair of profiles can be computed in linear time and gave an exact integer linear programming formulation for reconstructing a phylogenetic tree between copy number profiles with minimum number of events. These results rely on two properties. First, for any pair of profiles, there is a shortest transformation where all deletions precede all amplifications. Such a transformation with two phases of events is called an ordered transformation. The second property shows that in this transformation, the number of events that affect each position in the profile in each phase is bounded by the maximum copy number. The copy number distance has the disadvantage that it gives all events equal weight regardless of their length, position, or type, amplification versus deletion. This limitation has several drawbacks. First, copy number aberrations have been reported to have different rates in different cancers depending on their location, length, and type. Second, the copy number distance is sensitive to errors in the copy number calls, as each change is counted as a single event. The discrete nature of the copy number distance makes it hard to distinguish between small focal events that are possibly errors from large-scale events such as chromosome losses. Finally, for a pair of profiles, there may be multiple shortest transformations between them, and some of them may be more plausible than others. In this work, we propose to overcome these limitations by deriving a weighted copy number distance. To model differences in events, we introduce an event weight function that gives each possible event a positive weight. The event weight accounts for the position of the event along the chromosome, the length, and the type of the event. We define the weight of a transformation between a pair of profiles as the sum of all events in the transformation. The weighted copy number transformation model can distinguish between transformations not only based on their length, but also based on their weight. Going back to the previous example, if we assign a weight of 1 for deletions and 2 for amplifications, then the weight of the first transformation is 3, whereas the weight of the second is 4. It is not immediately apparent that the weighted generalization retains the combinatorial property of the copy number transformation model that enables its efficient computation. For example, while reversal distance between sign permutation can be computed in linear time, the weighted reversal distance is NP-hard. Both the linear time algorithm and the ILP for computing the copy number distance restrict to ordered copy number transformations, where all deletions come before all amplifications. However, in some cases, unordered transformations may also be biologically relevant. Since our goal in the weighted copy number distance is to find more plausible transformations, we want to look at a less restrictive solution space. In the previous example, we see that one transformation is ordered and the other is not. We generalize the definition of order transformations into semi-ordered transformations, having three phases: deletions, amplifications, and deletions. We give an ILP for finding the shortest semi-ordered transformations and characterize all possible transformations that can be generated from this ILP via maximum matching between event starts and event ends. We now turn to our main result, finding a minimum weight copy number transformation between a pair of profiles. The weighted copy number transformation model can also be interpreted as a probability model. Suppose that each event occurs with some probability and that events in a given transformation are independent. If we set the weight of each event to be its minus log likelihood of its probability, then a maximum likelihood transformation will correspond to a minimum weight transformation. We solve the minimum weight transformation problem by formalizing it as an ILP. But what we show is that the constraint matrix of this ILP is totally unimodular. Therefore, we can relax the integrality constraint and formalize it as a linear program. This linear program has an integer solution corresponding to a minimum weight transformation. Note that since the minimal weight transformation problem does not find the shortest transformation, it may produce very long transformations. Therefore, we may want to find a transformation that balances both its weight and its length. We propose two regularized versions for the minimum weight transformation problem. In the first version, we propose to balance a linear combination of the transformation weight and length. This corresponds to having a geometrical prior on the transformation length. In the second version, we propose to find a minimum weight transformation only among the shortest transformations. We show that both problems can be reduced back to the minimum weight transformation problem, therefore can be solved with the linear program. Finally, we show two applications of the weighted copy number transformation model. As a first proof of concept, we wanted to test in simulation whether using a weighted copy number distance with knowledge of the distribution of events gives better estimates of the evolutionary distances than unweighted copy number distance. We simulated copy number profiles from a directed tree via copy number events that occur with different probabilities. We draw the length L of an event from the following distribution parameterized by beta. We assume that the tree is unknown, but the probability of distribution of events is known. We further assume that all profiles, including inner nodes, are used to reconstruct the evolutionary tree and estimate the events along the edges. To reconstruct the tree, we first calculate a pairwise distance matrix D. Note that D is not necessarily symmetric, as both copy number distances are not symmetric. Moreover, for some ordered pair of profiles, there may not exist a transformation between them. We test three distance measures: the Euclidean distance, the unweighted copy number distance, and the weighted copy number distance, where we use the minus log probability of each event as its weight. From D, we build a directed weighted graph where we add an edge between nodes only if the distance between them exists. Finally, we find a minimum spanning arborescence in G. For the copy number distance, this corresponds to the maximum parsimony tree, while for the weighted copy number distance, this corresponds to the maximum likelihood tree. We tested different event distributions as a function of beta. First, we calculate the percent of correct edges between the true and inferred tree. We find that the weighted copy number distance shows significant improvement over the unweighted copy number distance for all values of beta. The performance improvement is bigger in smaller values of beta, where the events are longer and more likely to interact. We also calculate the difference in the total number of events along the edges of the true and inferred tree. This quantified how well the inferred tree can recapitulate the events of the simulated tree. We found that the weighted copy number distance infers trees with almost zero difference in the total number of events for all values of beta. On the other hand, the unweighted copy number distance obviously yields trees that underestimate the true number of events of the tree. Since in real data the true distribution of events is unknown, we repeated the simulations using different probabilities for tree infants with a weighted copy number distance and found similar results. In the next application, we analyzed copy number profiles derived from ultra-low coverage single-cell whole genome sequencing data of a breast tumor obtained using the 10x CNV solution. Chisel, a recent method from our group, was used to infer haplotype-specific copy number profile for each cell in 5 megabase bins across all autosomes. Thus, each cell has 44 copy number profiles corresponding to the 22 pairs of chromosomes. We analyzed cells jointly across all 5 sections of the tumor, focusing on cells that Chisel classified as tumor cells, resulting in a set of 4,000 cells by 1,000 copy number entries. Due to the ultra-low coverage, copy number calls in individual cells are prone to errors. While whole chromosome and arm-level copy numbers are more likely reliably called, focal single-bit changes in the profiles are more likely to be errors. The Chisel analysis addressed this issue by clustering cells, resulting in 8 groups of cells, or clones, labeled 1 to 8, and characterized by different large-scale copy number aberrations, including whole chromosome and chromosome arm-level events. The analysis also built a phylogeny relating these clones with 2 main branches of clone 1 and 2 and clones 3 to 8. Since the individual cell profiles are noisy, we first analyzed the clone profiles. We created consensus copy number profiles for the cells of each clone. We then calculated a symmetric distance matrix between clones and used neighbor joining to construct a phylogenetic tree. Since the major clonal events in the sample are large chromosomal aberrations, we gave whole chromosome and arm-level events a high weight. On the other hand, we suspect that focal changes are more likely to be errors and thus assign these events a very low weight. Both the Euclidean distance and the unweighted copy number distance misplaced one branch in comparison to the Chisel phylogeny. On the other hand, only the weighted copy number distance produces a tree concordant with the previous analysis. To show that the weighted copy number distance is able to cope with errors, we next computed phylogenies on the 4,000 individual cells using neighbor joining. The tree based on the unweighted copy number distance mixes cells from different clones in the same clade and even cannot separate the two main lineages of clone 1 and 2 and clones 3 till 8 that are most distinct in their copy number profiles. This is likely because the unweighted copy number distance is susceptible to noise in the copy number profiles of individual cells, and small differences in the copy number profiles are weighted equally as large events. Conversely, the weighted copy number distance infers a tree that separates the two main lineages, preserves most of the clonal structure, and recapitulates the evolutionary relationship from the chiseled clone tree. In this paper, we derive a weighted copy number distance that allows for segmental events to have different weights or probabilities based on their type, length, and location. We develop an efficient polynomial time algorithm to compute the weighted copy number distance and find a minimum weight transformation. This is in contrast to other genome evolutionary models where the weighted version is NP-hard. We illustrate the utility of the weighted copy number distance in three applications: distance-based phylogenetic tree reconstruction, analysis of noisy copy number profiles from single-cell sequencing data, and estimation of copy number aberration rates in pan-cancer data, which I refer you to the publication for further details. An important question in the application of weighted copy number distance is how to determine the weights. We showed that it is possible to estimate some copy number aberration rates in cancer cohorts, but there is still room for improving this process. Another direction is to extend the probabilistic model for copy number evolution to account for dependencies between events, modeling errors, and even more complex mutations. I would like to thank Ben Raphael and members of the Raphael Lab, and of course you for your attention.